Welcome back, everyone. After we have learned that replication itself is semi-conservative, let us have a closer look at the details of this process. So, how are nucleotides added in DNA replication? And in order to understand this, you need to remember that DNA molecules do have polarity. So our DNA strands have a 3' and 5' end. And please do keep in mind that the strands within the DNA are antiparallel. That means that the newly synthesized strand is gonna have different polarity because the 5' end is gonna be here and 3' end gonna be here, in contrast to the original template strand. If DNA polymerase is adding nucleotides, this only occurs at 3' end, right? So this is important. New nucleotides in the new strand are only added at the 3' end of the new strand. This is because polymerases can only read DNA sequence in 3' to 5' direction. So in this one, but they only synthesize in 5' to 3' direction, right? So this is the 5' direction and this is the 3'. So they are reading information from here to here on the template strand, but they are synthesizing from 5' to 3'. Okay, and this is something very important and something that you should keep in mind. In addition, DNA polymerases require a free 3' end to initiate the synthesis. So what you have actually here is a free 3' end. Again, as I mentioned before, DNA strands are antiparallel. And this actually causes a problem for replication. So what problem might this cause? So let us have a look at the strand of DNA. As you can see here, this is a normal DNA molecule containing of two different strands. During replication, obviously those strands need to open, and due to antiparallel nature of those molecules, a new DNA molecule can be synthesized perfectly well on one strand, and we call this one the leading strand, because here DNA polymerase can do its job. It can read from 3' to 5' and synthesize from 5' to 3'. However, on the second strand, the so-called lagging strand, this is more difficult. Keep in mind that DNA polymerase can only read in one direction and produce in one as well. So here the polarity is different. Therefore, DNA is synthesized in a discontinuous manner on the lagging strand. Okay, so DNAs can produce the full molecule here, but it needs to stop at the multiple points on the lagging strand. So how does replication look like step by step? So the first step of replicating DNA, of generating identical copies, is obviously opening the molecule. And this is the job of an enzyme called helicase. Helicase untwists the double helix and exposes complementary strands at the origin of replication at the so-called replication fork. This is the place, the DNA, where replication actually starts. Afterwards, another enzyme called primase puts down short piece of RNA termed the primer. And this is actually quite important because those primals generate free 3' ends, allowing the DNA to work. Primers are existing group of RNA nucleotides with a 3' hydroxyl group to which a new nucleotide can be added. And they are usually 10 up to 12 nucleotides long. Once this is done, our DNA polymerase can read the template strand from 3' to 5' along each naked single strand, adding complementary nucleotide to the new strand. And again, the process is slightly different between the leading and lagging strand. On the leading strand, the DNA synthesis is continuous. However, on the lagging strand, we have the primers that bind to different sites of the DNA. However, the DNA cannot synthesize the DNA in a continuous way, and this generates so-called Okazaki fragments, short fragments of DNA. So during the replication, each nucleotide strand serves as a template for building a new complementary strand. So in replication, both of the original strands are getting identical copies as a result of the process. So what happens after DNA synthesis has been complete? So once this is done, we still have a problem because our RNA primers are still present. And they need to be replaced by DNA in order to obtain a full new identical copy. Those primers are removed by another enzymes. They are not going to be talking about today. You don't need to know it as a pharmacist. But after those are removed, there is still a gap because those RNA primers are removed. And another enzyme called ligase identifies those nicks in the DNA and seals those and closes the gap. After this is done, replication machinery stops at specific termination sites, and this completes one round of replication. So what are the steps of replication in a nutshell? Again, as I mentioned before, we have the initiation, where the DNA strand opens. Then in the initiation, DNA gets prepared for complementary base pairing, and the next step is elongation. During elongation, proteins connect the correct sequences of nucleotides into continuous new strand of the DNA. After this is done, termination occurs where proteins release the replication complex. This is basically what happens during replication. So what are the basic rules of replication? And I think this is something you should keep in mind as well. First of all, replication is semi-conservative. In addition, it starts at the origin. Synthesis takes always place in the 5' to 3' direction, and replication is semi-discontinuous. Because it generates Okazaki fragments. And those are the things you should certainly keep in mind. In addition, RNA primers are required for successful replication. So let's have a quick question on the content. So, which of the following is true about the DNA polymerase? Oh, I already spoiled the answer, so I'm going to put the form slightly ahead of this. So obviously, answer A is correct. It can synthesize DNA in the 5' to 3' direction, and this is quite important. All of the other answers are basically wrong. And this is actually quite useful for your exam as well. If you have to deal with MCQs, and if you're not certain what answer is actually correct, it can help if you try excluding other answers that are obviously not correct. In this case, because we have learned that DNA polymerase produces DNA, we can exclude C, D, and E. Because, well, those answers claim that DNA polymerase synthesizes mRNA, so we can easily ignore the rest of those answers. This reduces the number of answers to 50-50, giving you quite high chances of basically answering this question correctly without even knowing the answer. Good. So, why is replication important for pharmacy and pharmaceutical industry? Because there are many drugs acting on replication. For example, the antiviral drug acyclovir. Acyclovir can be purchased as an ointment. You can also get this as a tablet. I think that the mode of action is really, really brilliant. This drug is used as a key drug against herpes viruses, CMV, and so on. So how does it work? The mechanism of action is basically that ACV, a prodrug, is activated by phosphorylation. Once this is done, it mimics the structure of GTP, guanosine. And again, guanosine is used during replication. So what is quite important, GTP actually does have a free 3' end, allowing replication. However, acyclovir doesn't have it. So it is able to bind to the DNA, but it stops replication. And because it is more specific to the viral replication mechanism, is it quite useful as an antiviral drug. So now I would like to show you a quick video summarizing what we have learned about DNA replication.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite 3' to 5' orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
With this, I would like to end session number 2, and I will see you in the session number 3, dealing with the process of transcription. Thank you very much for your attention.